to the Krebs cycle, then to the electron transport chain. So let's go ahead and start with our glucose molecule. We know that this is produced, glucose is the main carbohydrate that we are using for energy in our aerobic metabolism. And let's say that we're starting with 1 molecule of glucose. We'll come back to that. So after glucose, we know we go to glycolysis. Here is where the fork in the road happens, okay? Do we have oxygen or do we have no oxygen? That's going to determine where we go after glycolysis. So if we have no oxygen, we know that that is our anaerobic glycolysis that eventually ends in lactate formation. And we'll talk a little bit more about lactic acid formation, I think next week before we finish up Unit 2. So that's what we need to know about our anaerobic, no oxygen side of glycolysis. So if we have oxygen present, we know that we are going, oh, I don't actually want to delete that whole thing. I just want to make it a little bit smaller. Okay, so we know that we are going to go to pyruvate or pyruvic acid, depending on what version you want to use. So we know that pyruvate, just go ahead and write 2 molecules here. So we know that we have our oxygen present. We know that we're in pyruvate. We also know that pyruvate is 3 carbons. So we've gone from glycolysis to pyruvate, but now the main molecule that we want to get to is acetyl-CoA, right? None of this should be new so far. So acetyl-CoA is what is going to kind of prompt us to start the Krebs cycle. But before we can get to acetyl-CoA, we need to actually break down this pyruvate molecule. And so what happens is we go from pyruvate, which is 3 carbons, to acetyl-CoA, which is 2 carbons. And this is done in a chemical process here. I'll put some dots to sort of explain this chemical process that's going on. So we know that we are going to lose a carbon. We're going to lose a carbon molecule. So we're going from a 3 pyruvate carbon to a 2 carbon acetyl-CoA. Okay, so we're going to lose a carbon, and we're going to actually lose this carbon as carbon dioxide. Okay, so that's important for this. So as we go from pyruvate to acetyl-CoA, we also have some electrons that are going to be transferred to produce ATP. So we're going to have some electrons that are transferred to NAD, to NADH to produce ATP. Okay, so we're going to have to use a little bit of energy for this process, but we will produce some ATP off of that. So hopefully you understand how to go from our 3 carbon pyruvate to our 2 carbon acetyl-CoA. Know that through that chemical process, we are going to have some electrons that are going to be transferred to NAD to produce NADH that eventually is going to produce a little bit of ATP. I'm going to put a different color here, and just to remind you that through glycolysis, we are going to produce 2 ATP at this step. So if you want to kind of just write 2 ATP sort of up here with glycolysis, we're going to add up our ATP at the end. Okay, so I'm going to get a little bit of a better start here. So we have acetyl-CoA. We know that the main substrate that we are going to use to get to acetyl-CoA, we know that we can get there through carbs, specifically through glucose. But if you remember on Monday, we can also get to this step of forming acetyl-CoA from fat metabolism. And we can also get to this step of acetyl-CoA from protein metabolism. We are going to spend a lot of time on Friday talking about fat metabolism or lipid metabolism. So I want you all to understand that since acetyl-CoA is sort of our signal to start this Krebs cycle, know that we can get to acetyl-CoA from kind of 3 different ways. Okay, so let's remember that acetyl-CoA, we have 2 carbons. Okay. So now this is the signal to us to start the Krebs cycle. Okay, acetyl-CoA is going to start our Krebs cycle. So, couple terms here. We sort of introduced the term oxidation. So oxidation of acetyl-CoA simply means removing electrons to ultimately produce energy. So that's kind of telling you automatically what the Krebs cycle's purpose is. So we know that the Krebs cycle is a series of 8 chemical reactions. We know that our goal is to produce ATP, also NAD and FAD to transfer our electrons and the associated hydrogens to the electron transport chain. So acetyl-CoA, I'm just going to write it again, is 2 carbons. And we know that acetyl-CoA, it's going to combine, acetyl-CoA is going to combine with oxyl acetate. Now, oxyl acetate is a 4-carbon molecule. Okay, so the 2-carbon of acetyl-CoA plus the 4-carbon of oxyl acetate is going to form citrate, which is going to be 6 carbons. 2 plus 4 equals 6. So now we are going to continue along with our Krebs cycle. So we said that this is about 8 to 10, I think our textbook says 8, so chemical reactions. To produce ATP, NADH, and FADH. So we want to produce this ATP, NAD, and FADH so that we can have these energy-rich electrons that are going to go down into the electron transport chain. So let's kind of add a little bit more detail to the Krebs cycle to help us understand where these hydrogens and associated electrons are coming from, in particular, how many NAD and how many FADs are formed. So after citrate, we are going to have the chemical reaction to get us to isocitrate. After isocitrate, we are going to go to keto glutarate. Now, during all of these chemical reactions, and we'll write out a few of these, and I'm trying to keep this sort of as simple as possible, but enough for you to obviously understand what's going on. But at each, particular this reaction, and I'll go through each reaction, we're going to give off CO2 here. But as we go from isocitrate down to ketoglutarate, we are also going to have NAD, and this is going to be our NAD plus, adding in our hydrogen, which is going to be NADH. So now we have the formation of our first NADH. Okay, let's continue on. After ketoglutarate, we've got our, I don't even know how to say this one. I'm going to draw a few more boxes over here just to help us continue along. We have succinate. So going from succinyl-CoA to succinate, we are also going to give off GTP at this reaction. And I introduced this term a little bit on Monday, and we know that GTP is a high energy substrate. That is going to help us whenever we want to form our ATP. Now, from our ketoglutarate to our succinate-CoA, we are also giving off CO2, and we are also forming new NADHs at this reaction as well. So there's our second NADH, and we know that these are carrier proteins that are going to carry our hydrogen and the associated electrons down to the electron transport chain. So after succinate, we are going to go to fumarate. Now, this is where it gets interesting. Instead of CO2 and NADH, we're actually going to produce our first FADH. So let's highlight this in, I don't know, green to distinguish it being different than NADH. Okay, we're almost there. Then we're going to go to malate. We're actually going to pick up some water here to help this reaction go. And then finally, malate is going to go right back to oxaloacetate. So oxaloacetate starts this reaction after acetyl-CoA, and the Krebs cycle also ends with oxaloacetate. So at this juncture as well, we have another NADH. I'll give you guys a second to make sure that you have this copied down. So this is our Krebs cycle. We have our 8 reactions. 1, 2, 3, 4, 5, 6, 7, 8. And we know that each of these reactions is going to give off some sort of NADH or FADH. So from our color codedness here, each turn of the Krebs cycle produces 3 NADH and 1 FADH.
Dr. Schmidt, oh, sorry. Is it FADH or FADH2?
So great question. So I believe that this one is actually the FADH2. I was trying to keep it simple because I didn't want to confuse you. So I don't need you guys to really, at least for our class purposes, delineate between that, but you are correct that I should distinguish between FADH and FADH2. But I'm keeping this simple, so just, you're good. Just know at that point, yeah, it's the FADH2. But yes, great question. Thank you. So I think that I sort of spit that out on Monday, that each term produces 3 NADH and 1 FADH. And it's like, where, how did that happen? So hopefully now from this, you can see that. I see a few blank stares. This is good. We're good. We got this. We got this. Okay, so each turn of the Krebs cycle is going to produce 3 NADH and 1 FADH. So the purpose that we said of this Krebs cycle is to remove hydrogen and the associated electrons to go to the electron transport chain. Now, a little bit more about FAD and NADH. So we know that FAD can accept 2 hydrogen, and we know that NADH can only accept 1 hydrogen. So just keep that in mind as we sort of move forward. So remember how I had you all put 2 ATP for glycolysis? Now I want you to put 2 ATP for 1 turn of the Krebs cycle. I also had you all write 1 molecule of glucose. And I said this a little quick on Monday, but know that 1 molecule of glucose equals 2 turns of the Krebs cycle. Okay, we're doing good on time. So I'm keeping this simple because I want to keep it simple, and I want you all to make sure that you understand the reactions that we're producing NADH, that we're producing FADH, what the goal of the Krebs cycle is, how many chemical reactions are in the Krebs cycle, how we even got to the Krebs cycle to begin with. So this is sort of, you know, going along the lines of a study guide. If you all can sort of write this out from memory and sort of understand what's going on at each of these different sites, then you will be good. So hopefully this is a little bit more helpful than me just, you know, reading paragraphs off of a PowerPoint, which I know is not a good way to learn. So I'm hoping that this is going okay for you guys today. So now we have finished the Krebs cycle